In this video, we examine the effects of cruciferous extracts such as I3C, DIM, and sulforaphane against cancer. As always, these ideas are discussed in more depth in the book Cancer Self-Defense 101 Quick Tips to Help You Survive. All citations for studies quoted in this video can be found in the book's bibliography at cancerselfdefense101.com. This video is intended as information, not medical advice. It is not intended to treat, diagnose, or cure any disease. Any medical advice should come from an appropriately credentialed medical professional familiar with your unique circumstances. Cruciferous extracts have a long history of use against hormonally driven cancers. The first recorded use of a cruciferous vegetable against cancer was by Cato the Elder, a Roman statesman, who wrote around 200 BC that if a cancerous ulcer appears on the breast, apply a crushed cabbage leaf and it will make it well. Hormonally driven cancers include breast, prostate, endometrial, and ovarian. Even brain and esophageal cancers have shown evidence of being stimulated by estrogen. Cruciferous extracts have been shown to denature, render harmless, the most dangerous form of estrogen, estradiol, which is known to cause cell and DNA damage. There are three forms of estrogen: I3C and DIM also inhibit aromatase, an enzyme that converts testosterone into estrogen. They also desensitize the main estrogen receptor alpha. 80% of breast cancers are estrogen receptor positive, meaning estrogen fuels their growth. So you may think that consumption of I3C may help fight breast cancer. They certainly do help prevent breast cancer. Studies have even shown that I3C increases expression of the BRCA gene, a tumor suppressor gene which is often underexpressed in breast cancer. However, consumption of I3C can actually lead to decreased bioavailability of tamoxifen, the standard of care drug for estrogen receptor positive breast cancer, by about 40%. This is because I3C upregulates CYP pathways in the liver responsible for oxidizing drugs such as tamoxifen. DM is a metabolite of I3C, meaning that the body converts I3C into DIM. Since I3C/DIM performs many of the same functions as tamoxifen, it may be tempting to wonder if I3C/DIM could replace tamoxifen. However, there are nowhere near the number of human studies on DIM for cancer efficacy as there are for tamoxifen. In fact, we are not aware of any clinical studies that have examined people taking DIM as a primary treatment for breast cancer. Additionally, although I3C can be found in cruciferous vegetables, there have been no studies that have shown negative side effects from consuming cruciferous vegetables concurrent with tamoxifen. Perhaps this is because of the presence of other compounds, such as sulforaphane, in the vegetables, which do not affect the CYP pathways as I3C/DIM does. Cruciferous extracts have been shown to have many positive effects in human trials against many cancers that do not use tamoxifen as part of the standard of care, including triple-negative breast cancer. A double-blind, placebo-controlled trial in 98 men with prostate cancer found that consumption of a broccoli sprout extract led to reduced expression of the AMKR and ARLNC1 genes, both of which are implicated in prostate cancer development. Broccoli sprouts are very high in sulforaphane. 29 people with pancreatic cancer consumed 90 milligrams of sulforaphane daily for up to a year in the form of crushed broccoli sprouts in capsules, while 11 pancreatic cancer patients consumed a placebo. The 180-day survival rate of the sulforaphane group was 75% versus 57% in the placebo group. In melanoma patients, consumption of a broccoli sprout extract high in sulforaphane led to a decrease in inflammatory cytokines, an increase in decorin, a human protein involved in tumor suppression, and the group consuming the higher amount of extract demonstrated a slower growth of skin lesions. I3C, DIM, and sulforaphane have extensive preclinical evidence in cell lines and animal studies. For instance, one review of sulforaphane summarized over 170 studies, all since 1999, in various cell lines and animal models, and all results were encouraging. Cancer types studied included bladder, blood, breast, colon, gastrointestinal, gynecological, liver, lung, neurological, pancreas, prostate, and skin. For more information on cruciferous extracts, see the book Cancer Self-Defense 101, Quick Tips to Help You Survive.